My name is Ravi Gupta. I'm Professor of Clinical Microbiology at the University of Cambridge, and I'm the recipient of the 2023 Translational Microbiology Prize. So my talk this morning was around COVID-19 and the virus that causes COVID-19, SARS-CoV-2, and I was describing really our involvement in the early research efforts into this new virus. And we looked at SARS-CoV-2 from a number of different angles. For example, we documented how well it was attaching to cells and how well it was infecting cells, as well as how the immune system was dealing with the virus and how vaccines were generating immune responses in our bodies to fight the virus. We studied a whole series of elements in conjunction with genomics and understanding how the virus was evolving, and put all that together to try and inform public health initiatives, especially as we started seeing variants of concern with lots of mutations arising. When I graduated from med school, I wasn't really sure about what I was going to do and sort of which specialty I was interested in, and I think that traveling had been an interest of mine, and I'd lived in different places, and so emerging infection threats was certainly something I was interested in, and HIV was the obvious one at the time. It was a pandemic still, and it was treatments were only becoming available at that time, and much of Africa was still denied access to treatment. So during the time that I was a young doctor, I saw and witnessed the scale-up of antiretroviral therapy, and then got interested in how the virus was going to respond to those drugs, and indeed it became resistant to many of the drugs that we have. So that's a sort of my entry point into understanding evolution of viruses in the face of treatments that we're providing. So those are sort of laid the foundations for me learning about not only public health but also molecular biology in terms of understanding how the mechanisms of this were working at sort of single cell level and single virus level. And then we got interested in, of course, how HIV persists in cells, how it becomes impossible to eradicate and cure. And then we eventually led a study into the London patient who was cured of HIV by stem cell transplantation. And that sort of set up a whole translational sort of angle to my work. And so when SARS came along, we had to apply lots of the virology we'd learned. We had to apply understanding of evolution to this new disease. And as a clinician, an active clinician, I also was seeing patients in the hospital and then asking the question of how are we going to tackle this infection in a patient more effectively, and how we're going to protect others by vaccines, and what's the virus going to do in response to that, and evolve to overcome vaccines and drugs. So it all kind of tied together with SARS-CoV-2. So it's really important to understand the evolution of this virus and actually what evolution actually means in terms of what the virus becomes capable of doing or how it changes its behavior. And that was really highlighted by SARS-CoV-2 because in the beginning, scientists such as flu virologists were called in as the most expert group of scientists, and they predicted that SARS-CoV-2 wouldn't really evolve very much, and it would be quite straightforward to deal with once we had a vaccine. But actually what happened was the opposite. The virus evolved at much faster rates than had been predicted because it had aspects of its biology that hadn't been understood previously. And this allowed the virus to evolve at much faster rate within individual people during chronic infections. And so the studies that we performed along with others tried to understand exactly what biological characteristics the virus was gaining. For example, increased infection of cells, targeting different parts of the body, for example, moving from the lungs to the upper airways. This was the case for Omicron, and we think that that's what made it a little bit milder in terms of the amount of sort of hypoxia or oxygen deficiency that people had when they came into hospital. So we were able to use biology to explain and understand what was actually happening in real time. And if we hadn't done that, then we wouldn't have been able to explain or actually tackle the disease in the way that we did. The big takeaway, I think, from today is that nature is throwing new challenges at us all the time, and with improving technology, we have the chance to understand them quickly before people get sick and die, and therefore we can actually make a big difference in terms of saving lives through applying basic science to new threats. And the really important thing here is that you can't just use sequencing to identify new threats and track them. You actually have to do the biology that lots of scientists have been doing for many years on different viruses such as HIV and flu, and apply those to these new threats. And so if we support basic science, we can actually have a really positive outcome on these new viruses that are going to emerge as we go forward. It's a huge honour to receive a prize, especially looking at the other individuals who have been awarded this in the past. I think that everyone's a really sort of impressive and leader in their field. And of course, as they've mentioned, these are awards to one person, but actually they represent, I think, awards to groups of people who we've worked with, my team members, and also science is really collaborative at the moment, so it's also an award, I think, for everyone we've worked with in the past.